Good morning. It's a great pleasure to be here. First of all, I would like to thank the organizing committee for the nice invitation. So I'm going to share my screen. So I'm from Lisbon, Portugal. I'm from a school of engineering. So actually, I'm the coordinator of biomedical engineering master degree. So you are all invited if you want to come and visit us, for instance, by an international agreement between both schools. So today I'm bringing a theme that is how to discover biomarkers for disease diagnosis, for instance, based on metabolomics, and why this is so important. So if you understand the disease mechanisms, you know that all diseases actually are subtypes of diseases with different types of mechanisms. For instance, on breast cancer, we have different types of breast cancer, and some of the types, for instance, as hormonal receptors. So they overexpress these hormonal receptors, and so the most efficient therapies is ones that are going to target these hormone receptors. But there are also other kind of breast cancers that overexpress other kind of receptors, and so the more precise therapy is not one based as the previous one. So understanding these various subtypes of diseases and discovering these biomarkers of these subtypes could lead to a more precise medicine. So on precise medicine, we want to subcharacterize all these diseases and find these biomarkers associated to these different types of diseases for a more efficient therapy. So, and how to discover these subtypes of diseases, these biomarkers of these subtypes? Well, actually, one of the most efficient and more innovative way to discover it is based on the omics science. That is looking at all the systems, at the molecular present on these systems, and try to understand what kind of difference exists between these different subtypes. And in the last 20 years, a great evolution, technologically speaking, has happened, and we are now able to do so. For instance, on the genomics level, on the last 20 years, when we started the Human Genome Project, we took around 30 years and many billions of dollars worldwide with lots of laboratories to design the first draft of the human genome. Actually, after these 20 years, we managed to do so much quick on a few days and a few $1,000 per genome. So we evolved a lot. But why genome? Why not the proteome? Why not the metabolome? So if you consider the central dogma of biology is like the information flows from DNA to the metabolites, as you are going through metabolites, you know exactly what the system is conducting. So for that, I'm focused on the metabolome at the end of this flow diagram. But considering the metabolome, what technique are you going to apply to acquire all the metabolites? We never know all the metabolites, but the highest number of metabolites are the most relevant metabolites on the system. Well, there's a lot of different techniques, one's more quick than the others, like nuclear resonance, that is in top of pyramid, but has low sensitivity when we compare it with the chromatography technique associated to mass spectrometry technique. This is a higher sensitivity, but are the most expensive, the most laborious, and so on. Even choosing the chromatography associated to the mass spectrometry, we are looking for different molecules. And all these techniques there are very time consuming. So actually on my laboratory, we are looking for a new different type of technique, like spectroscopy in the infrared region. On this area, we are looking for vibrations, bond vibrations in the different molecules. And even if in complex biological samples, we are not looking to quantify all these metabolites, we are acquiring a signature, a molecular spectrum that is like a signature of the molecular composition of our samples. Of course, we can conduct the quantification of the substance, but not many substances. Actually, we published almost 10 years ago one work where we quantify different types of molecules, but this is very hard working, implies a calibration process, even if the models are very, very good, because we have correlations coefficients near 1 when comparing data obtained by the technique and the other ones by conventional analytical methods. Even so, is not the best technique to be used here. Also looking at directly from the spectra, looking at spectral bands that are associated to particular molecules on the systems like RNA, DNA, proteins, lipids, and so on, we could predict apparent transcription rates and so on and so on. But even so, that is not the best way to acquire data. So what is the best way? One, I'm going to show one of our first example that is predict, for instance, genotoxicity of hospital workers from a drop of blood. So usually to understand the genotoxic effects due to exposition of the professionals to environmental factors, as for instance, drugs using chemotherapy, that is there's potential to imply genotoxic effects even on hospital works due to errors that associated to surface that are contaminated on the hospital and so on. To conduct this conventional analysis, usually we took blood from the professionals, and this analysis, we grow the cells outside in vitro, and then we're going to analyze the look of these leukocyte cells to see abnormalities on the chromosomes, on the nucleus. And this takes too long, it's like a week. So it's not a routine conducted test. And we add on this assay around 50 workers that were exposed, professional workers exposed to these contaminants, and other ones that were not exposed. And we look by this conventional assay that is chromosomal abnormalities increased on the workers that were potentially exposed to these drugs. So, but we want to know if we were from a drop of blood analyzed by infrared spectroscopy and based on a very small sample after very simple dehydration of our sample and acquire the spectrum in high throughput mode, because we are using microplates with 96 or 384 wheels. So we acquire the spectrum, we have here the spectrum or the second derivative spectrum at the bottom. And then we try to look by multivariate data analysis, like we could look for direct from the spectrum for specific bands. And we are comparing these two populations already see large difference between them. Or looking for the whole spectrum by principal component analysis. So we start to look patterns that are different between these two populations, the exposed in relation to the not exposed one. But using machine learning algorithms as support vector machines, for instance, and evaluating different modes to pre-processing the spectrum using derivatives or not, that increase the resolution of the spectrum, but also increases the noises. So we need to balance these two factors. We observe very good predictions to know if a person was exposed or not to this genotoxic substance. So the area under the receiving curve is almost 0.9, independent of the preprocessing method used applied to the spectra. So we have very nice sensitivities. So around 85%, we are predicting very well the people that are exposed to that already presents the genotoxic factors. So this was a very nice outputs. We are also applied this to other systems based on analysis of the serum, on this case on the leukocytes obtained from the blood from patients. So actually we want to know if the technique were able to know if were able to discriminate B from T lymphocytes. This is very relevant in many immunological disease to know what kind of lymphocytes population was activated, because in function of the lymphocyte that was activated, the therapy is completely different, and the routine analysis to conduct is like the previous one, very expensive, is not routinely conducted in every hospital laboratory. So based on these leukocytes analysis, taking the spectra again by high throughput, very simple mode. In this way, we look for a second derivative. Again, by support vector machines and evaluating different regions of the spectra or different, for instance, we have here the regions, the whole spectrum, not only a part of the spectrum. We know that, for instance, in this case is specificity very high, accuracy is very high, precision is very high. So we have excellent predicting performance. In this case was the signature associated to B and T lymphocytes was totally different. Even so, there are many questions about the technique. So we compared our technique on this particular case with patients from intensive care units, some of them under mechanical ventilations, other none, some of them deceased, other not. And so we compare all these subgroups with two different techniques. So we applied for all the samples, we acquired the spectra and the molecular profile obtained by the spectrum from all these blood samples. And from also the blood samples, we analyzed it by a conventional assay used to acquire the metabolome of the system. So in this case, with liquid chromatography associated to the tandem MES, so mass spectrometry. So, and we analyzed, for instance, based on mass spectrometry data, we obtained for these particular two populations that all of them were with invasive mechanical ventilation. So half of them deceased on the intensive care units. And I want to know, to predict before 1 week before the death, if you were able to predict the risk associated to a particular patient that is going to die or not. And this is very important for medical care, to know if this particular patient is going to worse, and so focus our medical attention to this patient to minimize this end. And we were able to obtain a set of metabolites, very identified by MES, and looking for this signature, we observed that the deceased patients 1 week before the disease present a completely different molecular profile. And so the machine learning algorithm in this case was a linear discriminant analysis. Based on these metabolites, we were predicting with high sensitivity, with high specificity, which patients were dying or not before what happened. So we compared this data also with the infrared spectra data, and we were achieving very similar prediction performance with very high sensitivities, like slightly higher even with the infrared spectrum in relation to the metabolites profile acquired by mass spectrometry with very, very high specificity and so on. So this gives you a very interesting predictive performance. We are applying this to other very different clinical problems, like predicting the rejection of the renal organ transplant patients before that happens, predicting bacteremia in intensive care unit patients. Well, we are running lots of different examples, but the last one that I want to speak about is about a completely different it's based on infrared technique, but not looking for the average spectrum, but looking for the image. So we are working with the microscope with the focal plane array detector. That is, we are acquiring, in our case, 64, we have a 64 for the 64 array. So we are acquiring for more than 4,000 spectrum in that are equally distributed in a defined area, in this case 150 micrometers for 150 micrometers. And if you are looking, in this case we apply it for the prostate samples to try to predict prostate cancer in relation to hyperplasia. That is not prostate cancer, but is abnormality already. And we want to do it automatic instead of what conventional diagnosis that is based on the pathologist's microscopic optical observation. So acquiring the spectrum and just to look at the area behind some of these different bands, we're able to almost reconstitute the optical observation. So this is interesting. Here we have the average spectra of, just for curiosity, of the cancer in relation to hyperplasia and the second derivative. So looking just for the average, we are not able to separate because standard deviations are very high. The variability among patients are very high. So we try to evaluate different methods for spectra preprocessing, looking after derivatives or not, different algorithms of machine learning to try to understand what is the best algorithm to obtain the diagnosis equivalent to the pathologist, and we achieved that one with a almost 97% accuracy as predicting which sample present cancer in relation to the conventional mode. So we achieve this with high accuracy, but on this case, we are acquiring not only the metabolome, that is the small metabolites, but also the spectra of the proteins, the spectra of the nucleic acids, and so on. So you could say that is one of the most information-rich and concise way to represent the whole omics, not only the metabolome, but also the proteome, the genome, and etc. And we are achieving very nice prediction models based on machine learning algorithms. So we could say the technique has a great potential to discover these biomarkers, to subcharacterize all these disease types, or conduct in an alternative mode a very quick diagnosis. So hope you enjoyed my presentation. If you have any question, you have here my email, and I will be pleasant to speak with you. So thank you very much, and have a nice day. Thank you.